time and again, we have been bowled over by the indescribable foreignness of other worlds, and I wanted to take a few minutes this morning just to talk about the idea of looking for life as we don't know it. So picking up where Dave left off, I would argue that looking for life as we do know it has been a terrific starting point as we've gotten our feet wet with astrobiology, especially for a planet like Mars. We're never going to find another planet more similar to Earth than the solar system. Mars and Earth had these thick carbon dioxide atmospheres, protective magnetic fields, silicate crust, at least some episodic water early in their histories around the time life was getting started here on Earth. And we know that up to 1 billion tons of rocks were likely exchanged early in the histories, and there's a possibility that we could have just kind of caught life from the next planet over, that life on Earth and life on Mars might be ancestrally related. And we've developed, as Dave mentioned, these really robust approaches for Terran life detection. We know how to look for these well-established, widely accepted identifiers of biologic processes. And because we're starting with what we know, it's a lot easier to interpret the data. We've got signals that can be very rich in information. And importantly, I think we have a much stronger understanding of taphonomy. What happens when an organism dies? You know, we might not find a dinosaur in the Rocky Mountains, but we don't have to because we can find a dinosaur bone. And the same thing is true with molecular fossilization. We can, even if we lose a bunch of functional groups, we can still identify what was once there. But as we expand deeper into the solar system, I think it's really important that we're thinking about these new targets and the possibilities for life there, and the ideas of developing tools and techniques that go beyond just life as we know it, I think are going to be especially important. We've got a whole bevy of new moons that we are suddenly able to target much more sophisticatedly with these new solar propulsion technologies, huge rockets like the SLS. And when we think about, what might the, say, molecular building blocks of life on a world like Titan look like, where water's not even your solvent? You've got these hydrocarbon lakes as seen in this Cassini radar image. We just need to think broader. So NASA has recently funded through its astrobiology program a team of investigators called the Laboratory for Agnostic Biosignatures, or LAB. And I just wanted to talk through a few examples of the things that we're working on to target these agnostic biosignatures. And just to be clear, when I say agnostic, what I'm really talking about is without presupposing any underlying biochemistry, any particular molecular framework. So not necessarily life that's even carbon-based, just thinking as broadly as possible. And it comes at an exciting time. This is a report from the NASA Astrobiology Strategy, was out a few months ago saying, this is a great time to think about research on novel and agnostic biosignatures, kind of how it is we look for life as we don't know it, how we can contend with the truly alien. So just to talk through a few examples, so one idea is this idea of chemical complexity. And as my colleague and lab co-I, Lee Cronin, likes to say, if you find a 747 on the surface of Mars, you may not know how it got there, but it probably wasn't random. And so a neat paper that he put out about 1.5 years ago was really asking, what's the simplest way we can construct a molecule from its parts, taking with the simplifying feature of duplication? Because you can get high molecular weight polymers that aren't necessarily complex. They're just repeating small subunits. And so as you can see with this example of biphenyl, you could simplify that into a construction process that just requires 6 steps. And you can start to explore chemical space, and you can see that simple molecules like glycine can be assigned an index of 4. Much more complicated molecules like ATP can be assigned an index of 21. So this is adding a new element or a new type of bond, a new feature. And what Lee's found is that there appears to be this threshold above which it's very unlikely that you don't have machinery, biological machinery associated with creating that molecule. And so that's not taking into account anything about what the actual chemical constituents of that molecule are. So Lee's got a terrific talk about this Thursday morning at 11:30 for folks that are interested in how his algorithms are coming along. Along similar lines, we're also working on this idea of molecular complementarity. So one of the most exciting things I think that's happened in the world of genomics is we've had miniaturization and sequencing technology. So now we have these handheld sequencers that can only weigh 85 grams and draw a watt of power. And for folks that create instruments for spacecraft, this is half the battle, just getting something down this small. So NASA is currently funding 2 teams through Matisse and Coldtech to develop nanopore sequencers to send into space. They've been tested in extreme environments down here in Antarctica, as well as off at the International Space Station. And one thing you might say, well, sequencing, that's looking for nucleic acids, that's not agnostic, And so even though these kinds of technologies might be able to be used for non-standard bases, potentially even informational polymers, what we've been most excited on my team is about this idea of harnessing the power of sequencing to explore complexity of surfaces. And so this is not necessarily looking for any life that has nucleic acids involved, but the idea is that you could take libraries of millions of randomly generated short oligos, so strands of DNA and RNA that are just 30, 50 base pairs in length, and they'll naturally curl up and have secondary and tertiary structures, and they'll bind to a wide variety of analytes. And so we're talking about peptides and proteins, small organics, inorganics like mineral surfaces. Of course, from the RNA world, we know RNA sticks to montmorillonite, and even metals. And they'll bind with the same sort of specificities and affinities as antibodies. And so the idea, if you can just imagine a simple sort of end member, say you have a simple little mineral grain versus a cell, and even the most primitive cell on Earth contains a vast amount of information patterned on its surface. If you were to incubate with libraries of millions of randomly generated oligos, some very, very small proportion of those would bind, and they'd bind, again, with these very high specificities and affinities. And if you've got a surface with a lot of binding sites, a lot of different analytes they could bind to, you'll have many more diverse sequences detected versus something simple like a repeating crystalline structure. And because these reporters that we'd be using are nucleic acids instead of proteins, we could amplify them up using the PCR, polymerase chain reaction. And we think this could have a lot of potential, especially for ocean worlds where chemolithotrophic life might be much, much lower biomass than we're expecting here on Earth, where we have photosynthetic life. So related talks, exciting ones, just following this session in the Agnostic biosignatures session by lab co-Is Andy Ellington and Eric Anslin at UT. Those are going to be in the Cedar Ballroom this morning. We also have just this idea of compartmentalization and chemical fractionation. Can we find separation from an environment as an agnostic biosignature? So here are just some NanoSEMS images where you can see really different compartments can light up, and we might be able to look at this. These are the kinds of things that Chris House has been working on in his laboratory at Penn State, but we have lots of excitement about where this might take us. And then also energy transfer for extant life. This is this wonderful quote, "Life is nothing but an electron looking for a place to rest." So we've long known that microbes can utilize minerals as electron acceptors. They can use insoluble ferric oxides, for example. And what turns out is that the electrochemical properties of abiotic and biotic oxidation actually look different, and it's apparent. And even the simplest of setups, as shown here, you get this marked and sustained increase in voltage in these little reactors that have microbial communities that have been inoculated. This is every time iron sulfide tailings are added in this simple setup. So this is something Pete Gerges has been working on, and we'll be talking more about Friday morning. And in collaboration with Jay Nadeau and other co-I on our team, also this possibility of setting these specific potentials. So this last image is it's these lithographically etched glass slides, and you can look at these chips and you can set these very specific potentials, and you could cycle through looking to discover possible energetic niches for extraterrestrial microbes. So now some of these techniques, we need some time. These are very low TRL. Some of them are more at the concept stage. They need to be developed. But others are higher heritage. And so trying to think about how we can translate these concepts to actual missions, I think, is where the rubber hits the road. But one opportunity that I wanted to talk about that's pretty immediate is this idea of translating to, say, Mars missions. So MOMA, the Mars Organic Molecule Analyzer, is an instrument that's going to be sent on the ExoMars rover that launches next summer, will land the following spring. MOMA is a really fantastic instrument. It's a gas chromatograph mass spectrometer coupled to a laser desorption mass spectrometer. It's got this incredible range, not only for high molecular weight stuff, but also things that aren't easily volatilized. Two big advantages. MOMA and LDS mode can actually detect these heavy organic compounds even in the presence of perchlorates, which has been really stymieing. Every time we heat samples above 200 degrees Celsius with pyrolysis, perchlorates release these reactive gases that make it hard to, that obscure the signals, but MOMA can avoid this problem, and it also can analyze structure using its MSMS mode. So if you've got a peak that's of interest, a particularly interesting peak, you can zoom in on it and create its own fragmentation pattern. And so we've been doing ongoing work at Goddard Space Flight Center, which has been responsible for building MOMA, building this whole training set using these flight-capable instrument bedboards and flight prototypes to see if we can start pulling out complexity information, working in close collaboration with Lee on his complexity work as well. But this is just one example. Say here's this really interesting peak at 825 Daltons. We isolate it and we look at the MSMS pattern. We see these mass units that are repeating. They're separated by the same mass units. And so what this actually is indicating is that this is a polymer, which indeed it is. And if you saw a pattern like this, you would not think this is a complex molecule. But we're trying to explore chemical space and do this for lots of different things to build this training set, so that we can work with colleagues at the Santa Fe Institute on our project, trying to develop these machine learning capabilities where we can take this data and translate it into data processing techniques. So one thing I will say that's important to note is that these agnostic techniques are going to trade definitiveness for inclusivity. It's never going to be as definitive as, say, finding a hopane or a DNA sequence. But one thing that we're fortunate is when we send missions off into space, we usually get to send a package of instruments, not just one instrument. And so we've got a whole wing of our project thinking along the lines of computation and theoretical approaches, how we can start combining multiple techniques, data streams from that maybe lots of different biohints together, and using Bayesian networks, trying to convert these measurements into thresholds and likelihoods, trying to push away from this life or no life binary into, okay, this is 3 sigma away from what we might expect with abiotic processes. And I think Sarah's going to touch on some of these cool data interpretation strategies as well, so I'll leave it there. But I just want to say thank you so much to NASA for this tremendous opportunity, and we're part of NetEnfold, the Network for Life Detection, which is, I think Nikki's going to tell you about some events we have with Enfold this week that you're all welcome to join for. So I'll leave it there, and I don't know, are we doing questions or should we go on? Oh, thank you.
Do this first.
Should I go?